I will proceed. So this is the first of a series of outreach and training video seminars that we're going to be giving. We may give some of these multiple times that are not indicated here. So I'm hoping that this presentation, as it matures, will be given again in the next few months. The next meeting, which is to be determined date, is going to be by Bruce Arano and Kang Jin at Cincinnati Children's, walking through the top cell resource, in particular exploring COVID-19 immune responses. On the 23rd, we will have a more focused meeting. This is going to be primarily focused on people that need to submit data to the LungMAP ingest broker. So this is not going to be really a broad open community presentation. It's really going to be really focused on those folks that are submitting data sets and metadata to the ingest broker with the latest advanced interfaces that we've built in to make this as easy as possible to update and upload and curate your metadata. If you have questions whether this is appropriate for you, feel free to reach out to the folks on the call, or even enter some information into the chat. Ask Stephanie or Chelsea if you have questions about the functions of that. Later on in January, we're going to be giving a more detailed introduction. I'm going to have a very high-level introduction to how you explore LungMAP data sets in the LungMAP data browser and in Terra, in particular how you access FASTQ files or other files and use those to create harmonized results in the cloud. And in February, we are also going to be overviewing the redesigned website, which is currently being built, that is going to be a new facelift on top of the existing website with a mind towards an eye towards really making sure the website is as easy to use as possible and clear and obvious and not only accessible to LungMAP members and trainees, but to the research community in a very seamless, easy to use manner. So with that, we're going to be focusing today on some of the new features at LungMAP.net, focusing on primarily single cell genomics, but also on the LungMAP cell cards resource. So we're going to be going a little bit more deeper than we did a few weeks ago into cell cards in the Azimuth resource, exploring data sets in the data browser I just mentioned, and exploring data sets in the LungMAP.net Explorer interface, which will be fully live by next week. And then we're going to walk through some different scenarios and some specific questions you as a community might want to ask and hear about your suggestions. Again, please feel free to interrupt at any time. So I think I've shown this graphic a few times. This is a graphic illustration of what we think really LungMAP is. It's an ecosystem of tools. The ecosystem is not just tools developed by the Data Coordination Center, but really a community of tools and resources being developed at the different research centers, including very specialized tools for omics and imaging analysis and visualization or mass spectrometry. And so really there's going to be a lot of satellites coming off of this lower area in terms of the center portals, which we're going to be coordinating even further with the LungMAP members. And there are really probably at least 6 or 7 of these community center portals that are being developed, which we need to really support important links from all of the relevant LungMAP.net hosted data sets to those other data sets. Basically the 2 objects here are 2 hosted browsers from LungMAP.net, along with the LungMAP.net central portal, where metadata is really a separate area of the LungMAP DCC focus, which is getting data sets from the research centers, bringing them in to distribute to these different regions. Over the last couple months, we've made a number of updates, which include, as you'll see in red, new data sets, which are either static downloads or interactive data sets. In some cases, publications for certain resources, new resources that are being described here, website redesign updates. We have a LungMAP Omero server that is the public version is going online very soon. And then we have a number of new resources, including some of the ones that I'm going to be talking to you about today, including cell cards and the LungMAP Azimuth specific browser. So these resources are available from the front page, which I'll be going to in a moment. And I'm just highlighting which areas in particular we've updated and where you can focus on when you go to this website at lungmap.net. We've made a lot of updates to the single cell RNA-seq data sets there, with the goal of this really being a community browser of not only LungMAP data sets, but community large lung atlases, which are available to explore in a variety of ways. Similarly, we have LungMAP proteomics and lipidomic data sets with increased interactivity. These are part of the LungMAP cell cards paper. We have the LungMAP Azimuth and LungMAP cell cards resources, which are both linked to the cell cards paper that is going to be coming out soon, focused on redefining the predominant cell types in the lung in a highly curated, well-documented manner, and allowing for exploration of those cell types and associated images and data sets. Data sets are accessible through these links on the main page and through this resources drop-down menu in which you can select some of these additional resources right now. We're going to be beefing this up in different stages, including highlighting these community browsable data sets as new features on this website. So in terms of cell cards interactivity, as I'll go through in a moment, really the goal of providing this interface is multifaceted. One is to basically allow end users, the community, to really look at these terms and say, what is the preferred terminology that should be used in the lung community that's been adopted by LungMAP? And there are a number of parallel atlases that are being developed. So we want to basically, as much as possible, promote standardization and coordination of this terminology, definitions, and functional descriptions, markers, and links to things like the cell ontology. In each of these data sets, we're going to walk through the different features that you see here on the right. On the top, you're going to see links to these other resources that let you interactively explore data sets that have been harmonized relative to these annotations that are shown here by Yan Zhu's group. And you're going to see how you can get images that link out of here. For example, if you have a series of markers that are displayed at the bottom of this list, you have genes link outs that link out to this LungMAP graph database. You can access cell surface proteins and see what different entities exist in many, many different data sets in lungmap.net, including getting interactive querying of all relevant data sets with different marker antibodies, and exploring the distribution of those particular antibodies in both human and mouse samples at different developmental ages. So with that, I'm actually going to show you an example of this. So we're going to go to lungmap.net, which you're free to do as well. We're going to go to this cell cards option down here. It calls up this browser window, which showing the predominant cell types within or lineages within the lungs. You can open any of these up and explore more detail. In this case, we're going to go look at alveolar epithelial cells and type 2. And so if you click on type 2, you're going to get a set of definitions that come up. The first thing you're going to see is this identifier. Why do we have this identifier? It's just a unique way that if this name slightly changes in the future, this ID stays consistent. You can also see that this cell type also has a separate set of page at LGA. And so in this case, you have the LGA legacy definitions, positive marker signatures, negative marker signatures, transcription factors, ligands, receptors, associated data sets that reference this marker. And you also can query images based on time, based on specific markers within this data set and explore them within this image browser tree. You also have links to a resource called the cell ontology. So why is the cell ontology an important thing to link to? Well, it not only tells you where that particular cell type lies within kind of this hierarchy of well-established cell types, but it really lets you basically look for orthogonal data sets and annotations in different databases that are linked to this term. And so you really can get consistent standardized information for particular cell types across organisms, across tissues, and other data sets and species as you want with this consistent resource. And where there are terms missing here, and say some cases that we don't have an optimal term, cell ontology term associated with a term, we're working with the cell ontology to add that with curators that exist within LungMAP, both the DCC and in the research centers. So for each one of these terms, you have different numbers of annotations, which are provided to provide increased utility to understand what these cell types are, what the function of these cell types are, what the location of cells that are corresponding to the cell type, what is the experimental functional evidence that exists in the literature, along with literature link outs. You can click on these and go explore the source manuscripts, understanding the heterogeneity in cell states, again, with manuscript link outs, regenerative potential, links to disease, and again, other key references. There are also these markers here that are shown here. And so in this case, you have marker genes. If you click on any of these genes, or in this case, surface proteins or antibodies, you can get basically a link. You can explore where these markers are called out in different data sets, and what and explore their kind of interesting potential understanding within complex diseases, for example. So the first thing you see when you click on SFTBC is you see a definition of that protein. You have links out to these various resources. And then you have these different omics technologies or imaging technologies, which you can actually filter on. So if you want to, you can filter for subjects based on their age, based on the experimental assay that was performed to obtain that data, the sample type, LCM, sorted cell, slide, single cell, the research group, or the specific technology. And so in this case, we see IF, confocal, in situ hybridization data, proteomics, single cell genomics data sets, and bulk RNA-seq data sets, where this particular marker is listed as a key feature. If you click on any of these images here, you can get much more detailed information. So in this case, we're clicking on this one subject here. So this is a 4-month-old human. You have the protocol associated with it, and down here you have the donor details. So for example, you have D075, and we'll come back to that in just a moment. Those details are from Brindle. You can explore any of these images at a higher resolution. This is not the highest resolution. And so what we're doing here is we're linking right here where you have the image file. This is currently a TIFF. TIFFs are not the ideal images. So we're actually linking the full stack of images, of high resolution images that are going to be available in Omero. So when you click on this, you're going to get a link out to Omero, where you can explore this images with the individual channels at a high resolution, and even make your own customized views of these kinds of examples. If we go back here and you go down below, this is all information that is collected from the Brindle database that we're pulling in. If you have a particular sample, and it's anything linked, any objects here that are linked to, you can click on them, and you can also see what additional data sets exist for any particular sample. So in this case, for this sample, we have additional types of staining that was done on this sample that you can look at with different antibodies. We have specific RNA-seq data sets, in this case, that were included for bulk RNA-seq and computed tomography. And in some cases, there are single cell RNA-seq and other modalities that exist for a particular sample. So this is the browser, and you can do this type of browsing for any individual cell card's annotated cell type. But there are also these interactive resources, the top here, to explore omics data in more detail. And so for this, I'm actually going to walk back through the slides for a second to just introduce this data set and what this is. So in order to show a standardized data set for the cell cards, what really needed to be done is a really mass curation project, where we didn't just look at one data set, but we had used results that were integrated across many different studies. And so for this purpose, members of Yan Zhu's lab, in particular Minzi Go, integrated 259,000 cells from 72 donors from 5 published studies and 1 unpublished study from single cell RNA-seq data sets. These are all non-diseased, with adult and pediatric samples. The youngest pediatric sample is 10 years of age, both on 10x3', V2, and 3', or V3. For this analysis, Minzy integrated the data sets with this tool Bachelor, performed lighting clustering to identify the predominant clusters, and to find those the cell types within those clusters based on the marker genes that are really set out by the cell cards effort, with the UMAP coordinates generated by Monocle 3. Importantly, this is not the final version. We're calling this a version 0.1. This is effectively an alpha version at this point, with the kind of official beta going out in the next few months once additional analyses have been performed and optimizations on that data set. Importantly, Minzy is also working on a single nucleus RNA-seq version of this data set to optimize reference-based label propagation with this tool Azimuth, as we'll be talking about in a second, along with a fetal-specific and potentially a mouse-specific versions, which members of the consortium are all going to be working on together to generate and then host within an Azimuth browser and within this Shiny Cell browser. So Azimuth, the Azimuth browser is a specific type of technology. So Azimuth is a web browser based Docker container that is created by the Satija lab. And so the Satija lab is effectively, it has several components. It's an R Shiny app, which means that it's basically taking R apps and making them web accessible through a Docker container. And in this case, this app is allowing something for a method in Seurat called label projection. And so label projection is taking a reference data set and taking a new data set and projecting your data into that reference data set to find the cells that are best matching effectively kind of the equivalent of ortholog. So similar cells within your data set to the other data set to basically produce cell labels on that data set. The current browser can accept 10x Genomics H5 files, which are standard outputs of, for example, 10x Genomics. And the default loadout data set is a data set from the Witzik group from this Brindle sample D071. If you want to find out more of that sample, you can go to the lungmap.net database, put in D071 and see all the associated data from that browser. But we're just going to go to that browser right now. So if we go and click on the cell cards Azimuth link here, what you'll get is you'll get this interactive web browser that appears, which gives you the capability to either load your data set. So any H5 file on your computer, you just find it, select it, browse for it, and load it, or you can load a demo data set and view that. So what are we looking at right here? So we're looking at the repertoire of cell types that are described in that 0.1 version reference that Minzy created with nearly all, I believe, the lung and maybe all of the LungMAP cell cards, cell types, with the top 200 cells associated with that cell type listed as references for visualization. So if you click load demo dataset, this is going to now load effectively an H5 file that exists in memory. If this breaks, it will be interesting, only from the perspective that there may be more than 20 people using it at a time, but it will be interesting to see. In this case, there are 6,800 cells that are loaded in this demo data set. And you can adjust the filters here to say, I do not want more than, for example, 70% mitochondrial contamination. So we're going to get rid of those cells over here. You could also say that you want a minimum number of features or RNAs that are expressed in these cells for siege, and it will decrease your number. If you click on map cells to reference, at this point, the program is going to transform the data and basically align the cells within this data set to the reference from this H5 sparse matrix file. When it is completed, you'll be able to view where the cells are within the basically the UMAP embedding of the reference database. And there's basically a method, a PCA-based method to map the coordinates of the aligned cells to the reference UMAP embedding, and to explore your data sets in terms of specific markers in more detail. And so here we've eliminated a few cells in the process. If you go here on the left and you click on cell plots, you can see both the reference data set with all the cell populations. If you mouse over any of these cell populations, it will say what the cell type name is. If you are looking at the cell type variable, which you can change the variables to look at here, you can look at the predicted cell types now in your data set. So this is the reference example data set we loaded. There is an upper limit on the number of cells you can load into this Azimuth browser. And you can see what the frequency of each of these cell types are predicted in your individual data set. You can also explore this data set in more detail. And so if you click on feature plots, you see the expression of this gene, if it's a T cell specific gene, GNLY, within your data set. You also get it's an NK specific gene, my bad, within this data set. And you can see a violin plot for this NK specific gene over here on the bottom. You also can look for specific markers. In addition, you could put your own marker up at the top there and visualize that. Or you could say, I'm going to only look at this population of arterial cells, and I want to see the most specific marker that is predicted here. Select that, visualize again the specificity of that gene within this violin plot, and the expression of that gene within various different cell populations. You see in purple here, those associations. You can download these results. You can download the actual script that was used to generate these results effectively within R. And you can download a text file, which has just the predicted cell barcodes and the cell types associated with them. So if you click on that file here, this TSV file, you're going to get a set of both the cell types, those barcodes, the predicted cell types, the cell type score, and the mapping score that are associated with that. So if you wanted to do additional filtering on this data set, you could with this file. So that is the Azimuth browser using the reference data set that the Yan's lab created. But there's another way to explore. Is there additional questions on that?
Yeah, Nathan, this is Shing. I have a question.
Hi, Shing. Yeah.
Hey, so thank you so much. This is very cool. Can there be a manual? You know,
a manual for how to run this
through.
Yeah. I think
like,
that's a great.
How to set your 70% reference for the bar line for mitochondria, and what does that mean? Sort of thing.
Exactly. Yeah. So I think that in there probably actually is a reference that we should a manual for Azimuth that we should probably link to from the authors. So we can. I think that's a great suggestion for just including that on this page. So we'll go and find that PDF first and make sure we have a link to it.
Great. Thanks.
Sure. So there's another way to explore this data. And so this was the top 200 cells. If you click on this other link here, this is explore the single cell
Nathan,
RNA.
before you go there, so that was the demo data set that you did. So people can browse their own data set on that page.
Absolutely, yeah. So we could have easily just selected our own data set, and it would have had the exact same load time and similar results.
So that was the top button above that demo data set.
Exactly, yeah. So here, if you had browse here, you know, I have these sample data sets right here. So I have this 31-year-old 5' 10x genomics data set from the Witsit lab, which we could have also loaded in here and run this as well. Got it.
So is this needs to be a matrix file, or what is it?
Yeah, so the standard, there are two major standard files that come out of a 10x genomics workflow, but you can have other workflows that produce these files. So in H5 file, it's an and data object from Python, but R can also produce these H5 file objects. So these files can come out of the standard 10x genomics cell ranger workflow, or they can also be produced from other workflows like Seurat. If you have the other types of data are usually a .mtx file or a .mtx gz file. I don't think those are compatible with the Azimuth browser, but again, this is a standard output from Cell Ranger and from other workflows that do the alignment or pseudo alignment of the reads to the transcriptome, like Callisto bus.
Okay, and I expect this will probably be in the Azimuth manual, right? So
yes, exactly.
To have it for that reason too. So a question that you might have, you know, from the demos at GRC or other venues is that what happens to the cells that don't map to any of the clusters that in Minja's, you know, big map?
Yeah, so I think that, and this is a great question for the folks on the call that actually know a bit more about Azimuth than I do. But my understanding is that, that there aren't minimum mapping scores that exist within Azimuth. There may be. I'm not familiar with minimum mapping scores within Azimuth. So effectively what you'll get out is that file here, which if you effectively filtered on mapping scores here, you would be able to remove. So some of these values are very close to 0 and probably extremely low confident. You would effectively remove these low, poor mapping cells and either consider these as novel cell populations that are not in the reference data set, or just remove them for the purpose of downstream analyses that you're doing.
Got it. Okay. So I think that, yeah, we won't talk about it more, but I think that's a question that
sure, it's an important question, and I'll look through the Azimuth manual and see if I can address those as well.
Okay.
But pretty much every supervised reference-based classification tool for single cell has some usual minimum requirements for mapping, so that you don't believe certain things above a certain score. So it's important to know what score they have. It's likely above 0.4 or something like that you require is being specific, and that number is going to decrease with a new technology. So if you're using a single nucleus RNA-seq, which is literally low depth, you may get poor mapping scores for pretty much everything.
Yeah.
Xin, this is Yan. To add on the top of that, and so basically, the machine learning will help you to speed up, but I'm not recommended it totally. There's no reference now can be 100%, especially you focus on identify real cell type and the novel cell type. So you still do your unbiased the Levin clustering, and but it will save you a lot of time, like 90%. Maybe you can count on the reference, but still dig into the unbiased ones for questionable stuff.
So it's a great point to mention, and I did not mention it today, but in other talks I give, I talk about the concerns you should have when you do this analysis, and be wary of the results you get, because the results can be problematic for various reasons, similar to what Yan just mentioned.
And so Yan, since I have you on, so this 259,000 cells, does that include our eLife data set, the single nucleus?
Not in this one, but in the next version will be.
Okay, got it. Thanks.
Great. Any other questions from folks? I'll keep this open if other people have questions. So going back to the cell cards page, if you click on this Explore Single Cell RNA-seq option, another Shiny app will open up. You'll see the kind of similar Shiny app loading as you saw with the Azimuth browser. And this is now not just only the subset of the 200 cells, but I think this may actually be all the, is it 259,000 cells? Is that right, Minzy? For the whole data set. Sorry?
It's right.
Great. And so this is a published browser technology, which had a easy Docker to container that worked with a lot of our different variety of default files that are produced through our curation efforts already for LungMAP data sets. So we're initially supporting this, but we're looking into other technologies. There are different basically tabs here at the top that allow you to visualize the data in a variety of different ways. Along with produce different outputs here as PNG or PDF files, or change the types of visualization that you're looking at. So you can, for example, somebody can say that I do not want circles. I want to view the points as smaller dots, for example, or they want to they don't like this color schema, so they're going to change this color schema to a different color schema. So for example, yellow, green, and purple. You can order the cells within the plot in different ways to highlight different heterogeneity here. And so this is a nice set of flexible features for actually visualizing the data within these plots. And of course, this is, well, there's a default loadout gene here. You can replace this with any gene of interest that you would desire. And the loading for this is quite fast. So in this first plot, you can look at cell type information, which is a lot of different cell type information that's listed here, including numbers of features expressed, for example, in addition to any gene. You can just focus on not genes, but looking at different features. So looking at the number of features versus the cell type, exporting these plots out if you wanted to make a point for the paper you're working on. You can look pairwise and simply again look at the, pairwise, what is the expression of one gene versus another gene separately. You can also look at the co-expression of genes, and so you can see if two genes are co-expressed or expressed in very different cell populations by listing those genes here. Importantly, you can generate a number of plots here, which are quite useful, including violin plots, where you can look at either features or again, genes. So if we look at KDR, for example, you can get a KDR a specific violin plot. Again, you can there's a lot of ways to toggle these controls, select and restrict what you're showing here, and vary these plots before you export them. You also have these proportion box plots. In this case, we can switch these axes. So I'd prefer to actually look at cell types on this axis and actually look to see what's the cohort these are coming from. So from different data sets, you're going to have different frequencies of cell types captured, depending on where within the lung or the airway these different biopsies were captured. And you can also explore these data sets on a kind of on a broader perspective, using a group of gene sets that you can input in here as both bubble plots and heat maps, again, toggling these different visualization options to get different colors, different sizes of labels and things like that. And so we're supporting this kind of visualization for a few different data sets right now. We're still demoing and trying it out for different data sets, but we'll show you some examples of that as we go forward. But really, this is supposed to be basically an accessory to understanding these cell types, to explore omics data associated with these different cell cards terms. Before I move on away from cell cards, is there anything else I can answer? There's a lot of interface updates that we want to make here. Right now, there's no real description of what cell cards is. The name here is very basic. So these are all things that we're going to be updating, hopefully in the next week, as a very basic thing, and then adding more advanced features like image-based browsing capabilities, similar to what exists in LGA in the very near future.
Nathan, this is Xing again. So
yep.
The data sets that you just queried, it was the combined data set.
Yeah.
That Minzhe has generated, right? Was there a choice of other data sets yet? I know that was in the plan.
Yeah, so I mean, there's no other standardized data sets to cell cards yet, but what we'll be showing in a minute is how you explore other data sets with this. And I think we could have a kind of a more explore data sets in LungMAP, interactively explore, including, for example, the IPF Atlas, as a data set you can explore. So there's a number of community data sets with terms that are not aligned to cell cards that we can link to from here. Is that something that you think would be beneficial?
No, I think the combined data set is great, and to start with, and to show people, this is a rich data set that they would get a comprehensive query out of. But also mention that you are doing the data set, single data set based query. That will be sufficient, I think, at this point. But just one suggestion is to actually start the whole demo with that, what you just showed us. I think that most people coming to this, I'm attached to cell cards, but I am absolutely happy for you to push the cell cards much later, because most people coming to the demo want to say, okay, I want to look at this gene. And so you want to just even show people, here, type your favorite gene. This is how you do it.
Great. No, that's a great suggestion. Thank you very much. Yep. So here, I'm going to skip over this. We're adding a cell cards interactive feature within the Omero interface, which Josh went through before, and he's going to go through in a future Omero presentation. Then I'm going to skip over that for now and skip to other data set representations within LungMAP. So I'm actually going to go back to the LungMAP website right now. And I'm just going to highlight again that several of the new features are highlighted here under resources. And one of these new resources is the data browser. And so the browser is a tool that's been developed. This was initially developed hello. by the Human Cell Atlas. I was not expecting, this was working. Of course, they broke it today because we're demoing it. So that's the standard operating procedure. I'll see if this goes back online. I'm sure they're doing some fun maintenance today. Wow, okay. So I guess we're not going to be able to show this today. I'm going to try one more time to see if we can go to the data browser here. Oh, there it goes. And now it magically works. So apparently either I was hitting it too much or somebody else was hitting it too much. I don't know. But you have these different LungMAP data sets here that are located, that are listed here. Now we just have a few right now, and we've been focusing on the non-human data sets as we get our protocol for hosting human data sets here up and operational. And so that's going to be a few more months. But in the meantime, the non-human data sets are much more readily can go into this. You get this summary within this data explorer of how many cells are combined here. So this is focused on single cell genomics data, how many specimens, donors, how many files. In the extent, if you were to download all the data here, you'd be downloading close to 700 gigabytes of data. Now you can filter here extensively by donor in terms of age. You can filter by tissue type. Where's the, if we have anatomical regions that are very specific to the lung, you can filter by those. Specimen in terms of normal and different diseases. What is the protocol in terms of the library preparation method? What are the assays specifically that are performed or sequencing, or the particular file type? For example, count matrices versus FASTQ files. What are the data sets that have H5AD files, and so on. If you click on one of these data sets in particular, for example, you get a description of what is the title of the study, what is the description of the study. You can immediately download these data files or actually move to Terra with the data files to perform analyses. You see what the publications are that are associated. In these cases are preprints associated with the PI of the study, and citations along with the accessible geo-accession number and other files that are available. Importantly, if you go back to this page, you can always just browse directly by sample. So you can view all the samples here, select samples of interest. You can select all the files of interest. Again, the error, my gosh. We'll see if that goes away if I click. There you go, files. Now you get all the files, and you can take these files or from any study, and you can actually then go to, you can go to Terra with these files and perform additional analyses, or you could actually download the FASTQ files directly. So in the future, I'm not going to give a detailed evaluation of this. This is going to be a presentation that Tim and Trevor are going to be giving it in the near future. Any questions about that before I move on to exploration of individual data sets? So one of the primary features that we're going to see, and we really do need to highlight this in the homepage, and we're going to try to do that as soon as possible, are these new data sets that have been added to the omics visualization. And so 2 data sets were a part of the cell cards the paper, for example. Actually, I'm going to go over here. We can go, for example, if we go to lipidomics, and we see this interactive data set of this 20-month-old child. A very basic way to view this lipidomics data set is you can view it by within this heat map browser. And you can view it both in this heat map tree, highlighting the top molecules that Jeremy highlighted. You can also look at the mean expression of these different proteomic or lipidomic features. And you can do the same thing for the equivalent proteomics data set. For example, if we click on one of these donors, actually, I don't recall if it is the same donors. I believe it is the same donors, but you can access the other data sets that are the corresponding proteomics data sets, along with images from this same sample here. So again, if we look at the proteomics data set, you get the separate proteomics data set that you can compare to the lipidomics data set results, showing here the gene name, the protein names, or the gene names corresponding to the proteins that are visualized here within this heat map plot. And again, explore these in terms of their mean expression within the different samples that are assayed in this experiment. that is an example of proteomics and single cell omics data. But importantly, proteomics and lipidomics data set, but many of the people that are coming here are really now going to want to explore these large single cell atlases that currently exist. So there's a number of single cell atlases that we've been adding and supporting here. I'm going to walk through a few of them and show what some unique options are within these resources and tools to explore them and some more. So when you click, for example, on that Sun data set, this is leading to the Wang et al. Data set that was produced in eLife. You have the original eLife publication here, the LungMAP PI associated authors and first author. And you have this UMAP browser here, this interactive cell by gene UMAP browser. You have the individual sample file. So this is just showing the single cell RNA-seq and not the epigenome data. But if you wanted to access the epigenome data, you can click on this link to explore this over at UCS, the data at UCSD, or the link for this in LGA. And we're going to be adding a graphical link here, so they're highlighted for these different interactive features. You can also explore this by signature. So by default, there's these default loadout genes that are shown in one of the figure 1s from the paper, this Wang et al. Marker genes. But you can individually explore any intracell signature of interest here by selecting a cell signature, selecting search, and browsing for those genes, where each column here is a pseudobulk. So it's the average of cells from all the cells from one individual. And so the nice thing about this interactive heat map viewer here is you can select any value, you can get the individual value, you can see what the age of that individual was, what day, when some of these are children are 40, lungs are from a 3-year-old, 4 days old, 29-year-old, etc. You can see what the associated metadata here is in terms of age, donor, and other variables, and you can interactively explore the expression of these genes across age. So we can go back in a second, for example, and say, are there specific genes that are enriched in one particular cell population at one developmental time point? And you can come back here and you can view those genes, but the genes that are being displayed here are those genes I selected that were, I believe this was NK specific. So you're looking at NK specific genes here, and you're able to look at these by age. And you're also able to you are able to toggle these features in terms of what features you look at as the variables here associated with these cell types. I did want to just, show what is this UMAP browser is and what the capabilities of it are. So this is, very graciously, CZI is offered to host different lung data sets for us. And so for folks that's familiar with this browser, it takes a little bit of time to get used to it. But the important things here are these eye drops. So these are what you paint with. And so any of the variables you see over here, you can paint with those variables. So for example, and you can click open here to view any specific variables that you also might want to paint with. So for example, if we want to paint by cell type, you can view cell type. In this view, you do not see the labels automatically until you actually select this option up here, which shows you the labels in the graph that are most frequently associated with those different features. You can do the same thing for developmental age. So you can see, is there a skewing for different data points within different populations? Now, again, there's a layering. So some data points are hidden behind others here. You can view by ethnicity, since these are all samples with brindle identities. We know what the different associated covariates are. You can look by sex. You can also look by donor and by other variables here that happen to be present in here, including developmental stage. And so here you could say, you can do one very cool thing that I think is you can select a population of cells. So whenever you start drawing here, you can draw around any population of interest. And what you'll notice is down here, it says there are 4,100 cells. If I want to restrict those cells based on any of these variables, I can do that. So here we've got a 3-year-old, here we've got a 31-year-old, and here we've got 31-week-old. So if I want to compare what cells are in a 31-week post-fertilization kid, it says now are 2,600 cells, and I can select this as my first bin. So if I click on that, it stores those 2,600 cells. If I select off of here and I select the 31-year-old, I still have this selected. You can see there are 382 cells. So I select my number 2, and those are now stored. And this new option now appears. And if you select this new option, it's going to do effectively a differential expression analysis. And it says POP1 and POP2. So my first population here was the 31-week, and here's the 31-year-old. And so if we go and select this caret down here, and we select these genes, and we select that, again, that eye drop here, that paint drop, we should see the expression of genes which are preferentially expressed only within the neonate or the newborn. And so you can take any of these genes. And so, for example, if I want to take some of these genes, I'm going to just drag this over here, and I'm going to go make this window a little smaller, for example. And we're going to just look and see what's going on in terms of the expression of these individual genes. This is now where you can combine effectively these two types of browsers. So you can enter your own custom gene IDs to view in terms of the mean expression, where you can explore these in more detail. So you can see which are age specific by typing these genes in and getting that distribution plot in there. And this is a very effective way to do that. And it's something we'll probably highlight a little bit more later. I'm not going to go into the individual genes right now, but I just wanted to highlight that functionality. You can also search for specific genes here and view the expression of these genes again on this plot by having to select that teardrop. And so now you can see that SFTPC expression is expressed over here predominantly. And again, you could view this by developmental time points to see what the specificity are. So we've included this browser wherever we can within, go back here. Oh, actually, we had that open over here. Sorry, I'm just gonna navigate back. I move this. So we've added these types of features for various data sets now, and we're adding them for a number of more. So for example, if you click on this CRASNow data set, one thing you'll see is the UMAP browser that was not built by us, but it was built by CZI, which you can do the same type of analyses on, as shown here. You can link out to this top cell browser, which has the same data set. Again, you have this default loadout of marker genes, which are shown for the CRASNow data for the pseudo bulk of the different samples. And you can look at the mean expression of these individual genes, not only by cell type, but by region. So for in this case, it's location. So this is from a distal, this is from blood, this is from medial, this is from proximal. And so this is a useful way to actually view this data. In some cases, the cell types are only basically coming from one region of one basically potential source. You're going to get varying results based on that. We've also tried to support other additional community browsers. For example, we have the Kaminsky IPF browser that we want to link to, in addition to the interactive browsing of that data. But we want to make sure that you think of this as a shopping store where there's a one-stop shop to all these different portals that we want to basically give access to. One particularly useful atlas is this mouse developmental atlas from the Morrissey lab, which includes the same Shiny Explorer that we showed earlier, the same type of UMAP Explorer that lets you look at these developmental time points and do these comparisons, along with this heat map browser. And in this particular data set, what we've done is as the default view, we're showing not only genes that are specific to cell types, but are specific to time points. So for example, if you select a particular cell type of interest here, let's look at club cells. You can look at time point specific club cell gene expression, both within the UMAP plot and again within the mean expression plot, where again, these genes are really specific to club cells at a specific developmental time point. We really want to encourage these kinds of deeper exploration of these data where possible. And some of these require just going back through the original data sets and querying them in a different way to highlight different markers that are time point specific. This is the major features which we really wanted to cover today. And so I know we don't have a lot of time, but I'd like to give people the opportunity to ask any questions about capabilities or provide suggestions about what we can do better. Thank you.